Okay, so let's move to the next speaker, myself. So it's the first time that I'm a host and a speaker at the same time. So it's kind of interesting to introduce myself. So I don't know how much you want to know about me. I graduated from Fudan University in China and then moved to the US, did my PhD, and then moved to the Netherlands, did my postdoc there. And finally moved to Germany, start my own group in 2017. So I've been there for 7 years, and I've been always doing cross-linking for my entire scientific career. So boring and exciting at the same time. Since we're running over time, I can go a little bit faster just to skip some of the introduction slides because Manu and Yong had nicely explained cross-linking mass spectrometry technique. So cross-linking can be applied to purified protein complexes, organelles, and then the whole cell. And in our lab, we're mainly looking at cross-links from complex biological samples. So we do a lot of whole cell cross-linking, organelle cross-linking, and then the smallest biological system we've done is like a Varion, which has like 200 proteins or so. So then, as I said, I've been working on cross-linking for a long time. I won't tell you how old I am. So I think I started with when I was a PhD student, and then the cross-linking field, when people applied cross-linking to complex biological systems like protein-wide cross-linking. And back then, only like thousands of crossing could be identified. And then over the many years of the efforts, and I've been using we've been trying to develop different cross-linkers and different acquisition workflows, the software as well, just to increase the sensitivity and dynamic range of cross-link identification. As you can imagine, that we do whole proteome cross-linking, so we want just to see like the whole interactome of the cell in a single or multiple experiments. So the more cross-links we find, the better it is, and the deeper the interactome is from the sample. So just some quick overview of what I've been done so far. And I think in 2015 or 2017 or so, we developed a software called X-Links, but it's not X-Links in PD, it's X-Links standalone version. And then that software allowed the identification of crossings from proteome-wide sample without this limitation of the N-square problem. And of course, there were also many other works at the same time, like P-Link or XQuest, that solved this problem using non-cleavable crosslinker, but I was mainly focusing on MS cleavable crosslinker. And then around 2019, that we incorporated FAMES, which is a gas phase separation device that developed by Thermo. So we didn't really do much, but just the plug-in FAMES software into this is Orbitrap fusion mass spectrometer back then, and then we could see that the FAMES, just adding additional phase separation, can improve the crossing identification to 20% to 50%. And then shortly after, so we moved a little bit to the crosslinker development field, where we developed the membrane permeable and IMAC reintegrable crosslinker. We call it a T-butyl-FOX. And then this crosslinker, as you can see over here, that it has two. So on the left side is a FOX crosslinker, which was developed by Richard Schaltemer and Albert Heck's group. And there you can see they nicely build in a phosphonic acid group in the middle of the linker region, which allows effective cross-link enrichment using IMAC protocol. And IMAC is a really standard and popular protocol to enrich for phospholipeptides. But then since crosslinker has exactly the same group, which is the phosphonic acid, it can also increase enriched using this using IMAC protocol. And however, FOX is a great crosslinker, but it does not does not penetrate the cellular membrane. And the reason for that, we think, is because the phosphonic acid has two negative charged. Therefore, it prevents the penetration into the plasma membrane of the cell. So because of this negative charge, we thought about just neutralize the phosphonic acid with two T-butyl group and made the T-butyl-FOX on the right side of the slide. So you can see here that we just protected the phosphonic phosphonic acid with 2-T-butyl group, neutralize the crosslinker, making it neutral, and then this T-butyl Fox can nicely penetrate the plasma membrane of the cell. So now we have like a pair of crosslinker, both are enrichable, and the left side Fox is a non-permeable crosslinker, and the right side is a permeable crosslinker. So then we can look at the cell surface crosslinking as well as the intact cell crosslinking from this pair of crosslinkers. And then, and they're both available through Thermo Fisher Scientific, so feel free to buy them and try them out. And then shortly after, we also did some, just a small project a PhD student of mine did, which is just incorporating real-time libraries search, assisted crosslink identification, and there we just do like a short survey scan of MS2 scans to filter what are the MS2 spectrum, including crosslink signature peaks, and what are the MS2s do not have signature peaks from the crosslinker, and we kick the ones out without the signature peaks, and then do like a more comprehensive MS2 scans on the potential crossing species. And by this, we further improved the identification rate by another like 20 to 40%, depends on the different type of crosslinkers. So then what we're doing now, we're still developing a lot of tools to increase the sensitivity of crosslink. For instance, we're continuing developing softwares, new crosslinkers, and then we also do a lot of biological applications on using the crosslinking software that we've already developed. So as you can see that I have different people in the lab, and some are like relaxed. One says I gave up, but doesn't happen really often. And most of them says I want more, where I'm lucky that I find some really treasure from this gold mine. So in the next of my slide, I will show you some of our very recent work about like software development, because I think this is really nice, aligned with the topic of the meeting today. And then I also explain to you one of our recent biological applications on the very cross-linking. So generation of ground truth data set. So I think Manu and both Yong and Manu talk about the importance of a ground truth data set, because this allows us to compare different softwares, because we know what are the crosslinks are correct and what are the wrong ones. And then here we didn't do peptide because there were already people doing peptide, and then we switched to basically proteins. So here we expressed or overexpressed and purified 256 human proteins in E. coli, and then just each of them individually. And then so we have like 256 proteins in single Eppendorf tubes. And then we mix them in pairs, and then cross-link them in pairs, and then quench them in pairs as well. And then after quenching, then we pull them together and did the digestion and LCMS experiments. So in this case, we know exactly what are the crosslinks are correct, because the crossing coming from the protein pair sitting in the same tube for crosslinking, they are the correct ones. And then the crosslinks that coming from different protein pairs that we didn't mix them together, those are the wrong ones. So then we made this like ground truth data set, and then we use it to evaluate, to develop a software of our own, and also to evaluate the other available softwares on the market. So then the software we developed, we call it Scout, and then I think it just it uses the signature pairs. Oh, so come back, Scout only works with MS cleavable crosslinkers, because the non-cleavable crosslinkers, I think P-Link is doing it very well already, so I thought I wouldn't reinvent the wheel and do something that's even worse than P-Link. So, so then for non-cleavable crosslinkers, I would strongly suggest you to use P-Link. And then for the cleavable crosslinkers, let's see if you are convinced by my data or not. So, Scout searches non-cleavable searches cleavable crosslinkers by using the signature peaks of the after cleavage of the DSSO or DSBSO, just any other cleavable crosslinkers, and then it uses ion pair doublet searching. So it takes the precursor mass from the signature peaks and then perform the fragment I search. And so what is important, I think, for Scout is it controls the FDR at all levels. This is also what we put a lot of emphasis on, that we control the CSM, peptide pair, residue pair to the PPI level. So we just control, you can just put in, okay, I want 1% PPI level, and then it export to you the results from the 1% PPI level. So just some overview of Scout is an interface. It has a GUI, and then it has like a spectra viewer that you can see the annotated spectra. And then, so a very important slide, but also sometimes brings conflict. So it's a comparison of different search engines. Since we made this beautiful benchmarking data set, so we can actually use it to compare different search engines because, again, we know what are the wrong ones, what are the correct ones, so we can calculate the empirical FDR just based on our mixing scheme. So you can see that different bar plots represents different softwares, and Scout is ours, and then Xlinks, MS-Anika, MassLinks, and MerylX. So since DSSO P-Link hasn't published yet, so we couldn't compare to that, but I'm a bit nervous. I mean, if it's better, then it's better. So, so as you can see here, like similar to all the other observations, Xlinks has a really huge FDR, just because I also don't know why, but doesn't control the FDR very well. And then also, oh, I need to point out one thing is, so the dotted plot there says aggregated, meaning that the software does not report FDR, or does not control FDR at that level. So you can see that at CSM level, every software controls the FDR, but then at the residue pair and PBI level, then quite some softwares do not report a certain FDR at this level. So then we just aggregated the data. But this is not really a fair comparison, because if the software does not control the FDR at a certain level, you shouldn't say the FDR is higher, right? So then just to make it clear that for the aggregated data, that the software did not claim any FDR control there, so we just aggregated data. So it's expected that FDR is higher than 1%. But it is also important to realize from this slide that you have to control the FDR at every single level, because if you just aggregate it, if you just properly control the 1% FDR, the CSML level, and the aggregated data, then you still see like a huge explosion of the FDR at a higher level. So then what we also did here is just, we also compared the number of identifications filtered to 1% empirical FDR by post-processing. So here we did post-processing ourselves using the score cutoff, and then just to reduce the FDR to 1% based on our ground truth data. And you can see here that Scout still gives the best number, or the highest number of identifications at all levels when the FDR are controlled properly in all softwares. Okay, and then so this is a comparison of Scout versus XIFDR. We did it separately because we couldn't search the whole data set by XIFDR within a reasonable time window. So we just took a subset of the data and then did a search, but also took us probably like 2 weeks to finish just only a small subset of the data. It's just pretty slow. But although XIFDR is slow, but it performs really well. So you can see that, I mean, CSM residue pair doesn't matter so much, but you can see the PPI level that XIFDR controls the FDR pretty well, although it's slightly higher than 1%, but it's still among one of the lowest among the other software. And then the number of PPI Scout is only slightly better compared to XIFDR. And then, but this is a comparison filtered at 5% FDR. So you could see that Scout actually performs much better than XIFDR at 5%. So we're still trying to figure out why is it, but we do see a discrepancy at different FDRs for the performance of XIFDR. And then here is a comparison of the speed of different search engine. So you can see that Scout is really fast. And then also at the different sequences, the number of sequences in the database, from we used 4 different sizes of the database, like 400, 540, 2,000, 4,000, 20,000, and you can see that Scout does not perform much slower while the database goes larger. I didn't care so much of the speed in the beginning, but right now I really do, because like now we can generate, especially with Astro data, that's almost essential to have a fast software. Otherwise, like you generate the data and you couldn't keep up with searching the data. So I think with just, especially with the new Astro releasing, I think it's important to have a fast software. Okay, and then we also did a few comparisons using the existing data sets. For instance, the Max-Inger's peptide-based crossing standard, and then we just searched the standard using Scout, and then compared with the best performing software MS-Anika. So you can see here that we'll have relatively good identification, and slightly better than MS and Nika. And also, as what Manu said earlier, that the overlap between two software, the FDR is really low, and then the unique crossings for different software, for different softwares, the FDR is pretty high. So that's also a way of thinking about how to filter the data by just taking the overlap, the identification from different softwares. And then lastly, we did comparison of Scout versus XIFDR using the Lens PPI benchmarking cross-linking dataset. This is the one from Yuri Rapsoba, I think published last year or 2021. Yeah, just somewhere there. And then you can also see here, and here the overlap is lower. And we do see quite a lot of unique identifications from both Scout and XI search. And you see here that for both crosslink, for both software and then FDRs were properly controlled. Okay, so I have 10 more minutes, and I will give you just a very brief or quick biological application of the things we're doing in the lab. So we do a lot of cross-linking of the organelle, the cell synapse, the virion particles, various host interactions, because we finally have the tools to look at the interactome from large biological data sets. As actually the majority of my lab members are doing biological applications because it's really fun to dig into the data to see what's happening. So we have a lot of this interaction networks, so just connected by all the fuzzy balls there, and every single node is a protein, and the lines between proteins are protein-protein interactions. So you do see, so we have immense data on the interactome from different biological systems. And what we can do with the data, we can do a lot of things. So I will explain to you just one aspect of what we did with the cross-linking data. So the example I want to show you is a HCMV virion. It's a herpes simplex, like a cytomeganovirus, belongs to the alpha herpes virus family. And then, so it's a protein, as a virion with something around like 100 to 200 proteins, including the viral proteins and host proteins. So what is known about the virus? So the virus has the double-strand DNA as the inner core, that is the infectious material, and then the infectious material is embedded by a layer called nucleic capsid layer, which is shown in red here. And then outside of the nucleic capsid layer is a tegument layer, which is composed of large tegument, amorphous and flexible tegument proteins. And the very outside of the virion is the glycoproteins. The transmembrane proteins has a lot of glycan on it, which recognize the host proteins for the entry. So then what is known about the virion is the structures of the nucleic capsid proteins, the glycoproteins are known already, thanks to the high-resolution structure techniques, for instance, cryo-EM and cryo-ET. But what is not known is like the spatial organization and interactions of the proteins, the tegument proteins. And also, one very important question is here is the host proteins, is why the host proteins are inside the virus. So why, how they're recruited inside of the virus, and what are the biological significances of these host proteins sitting inside of the virus. So then we did cross-linking. And then we have a very complex interaction network. So you can see that in the middle region of this network is the tegument proteins. They're highly connect to each other. And also this tegument cluster connect with the nucleic capsid layer as well as the tegument layers. You can see it by colors. It's probably a bit too complicated to see here. So maybe I should directly point to the highlights. So one of the questions we're trying to ask is, we know there are host proteins inside of the virus, but we do not know where they are. So a question would be, like, if they're just randomly diffused inside the viral particle, or they follow a specific incorporation pattern and just sits at a certain location inside of the virus. So then what we did here is we just did a hierarchical clustering using the number of crosslinks. So then just made a heat map. So here this heat map corresponds to basically a virus, just to make it clearer. Like the very inner circle, that is the tegument, the nucleic capsid layer, and then followed by the inner tegument layer, outer tegument layer, and then the very outside of the layer, that is a glycoprotein layer. So you can see that from this heat map, the host proteins, they do not randomly diffuse inside of the virion, but a lot of host proteins, they only cluster, or they only connect to the inner, the nucleic capsid proteins, meaning that they are only specifically incorporated into that layer. And then we'll also see many other host proteins, they only crossing to the glycoprotein layer, so indicates that those are the host proteins only sitting at the glycoprotein layer. So by using this data, I just want to show you that cross-linking is able to capture the spatial location of every single protein in this network. And the reason is because cross-linker has a spatial arm of 10 angstrom. So then meaning that if you take a few proteins as seed proteins, like as localization markers, and then expand the network, you're able to figuring out what are the proteins connect to the no localization markers inside of the network. So yeah, so then, using this cross-linking data, we built like the spatial organization of every single protein in the virus, and then we also estimated the pairwise interaction probability estimated by cross-linking mass spec, and then we also performed this protein copy number estimation just by quantitative mass spectrometry, like a linear peptide identification, and then we also took available structures from PDB and AlphaFold models if there's no PDB structure available, and then finally we also took some sequence information so can predict whether the proteins have a transmembrane domain, and then we place them at the membrane layer. So with this, we made a quartz grain model of the whole virion that shows the location of every single protein, the interaction of every single protein, the copy number, and also like a quartz grain structure of a single protein. So that is to say that cross-linking can give you a lot of information, not only the protein-protein interactions, but also the spatial information. Okay, so I'm right on time. We'll have 5 minutes before. So this is my acknowledgement slide. I have 2 acknowledgement slides. So this is, so the virion work was led by Boris Bogdanov. He's a postdoc of mine, a great talented scientist, leading the work, and the Luda Verbish and Morrison Sadhihe. Those are the 2 great collaborators with the virion background, and then great expertise in quartz grain modeling. And then also my special thanks to the Scout team and Milan and Dio. Milan is a bioinformatician from Brazil, and Diogo is my informatician sitting in Germany, and then Max is just one of the students build the synthetic benchmarking data set. And I also like to thank ABC to providing me the 300 purified proteins. I actually didn't do it myself, but I kindly got them from a company which is also sitting in Beijing. And then we're hiring. If anyone is interested, just come by to have a chat. And thank you for your attention. Oh, right, I should host. So any questions?
刘帆，我想我的那个问题是一个 technical 的问题，就是你在做那个256个蛋白，把它们两组合起来做那个 benchmarking data set 的时候，这个每放在一起成 pair 的这2个蛋白，它们就是相互作用的蛋白，还是不一定有相互作用？
不一定是相互作用的蛋白，就是我们其实是属于 induce interaction， 就是放一块儿，然后就是完全的是 induce 出来的，就不是真的 interaction。
因为那个我们，哎呦，好早了，杨斌也在，就是这都是他做的实验。就是最早的时候，我们就测试过，把3个蛋白放到一起，就是完全不会有相互作用的蛋白。它那个分子，因为它们不应该有相互作用，但是我们把它放在一起，而且就是调它那个浓度哈，有的会放得很高，就是发现其实它那个交联很少，当然那个时候的仪器也不那么灵敏。
我们加热了，就是我们其实测了很多，比如说用很高的浓度呀，然后加热一下呀，然后加点urea啦，加热的会 induce interaction。其实加了urea或者 SDS 之类的，它会完全 abolish interaction， 就是因为我觉得你那个蛋白变成 linear 之后，它其实没有任何的 interaction。但是如果 induce 一些就是 aggregates 之类的，它就 interact， 所以完全是 artificial 的 interaction。但是它不太影响做 benchmarking， 因为我反正放2个，它就是一个就是我让它 interact， 就是跟 biological system 没有任何关系，但是它可以做一个 mass spec 的 benchmarking。就我们加热了。
这个我都不知道该怎么想。行，
其实跟 synthetic peptide 是一样的道理嘛。
我就是不知道怎么想，因为它是一个这个 artificial 的这个 condition， 不是 natural 的这个 protein interaction。
是的。
所以是不是一旦加热了，有这个高浓，就是你把蛋白放在一起加热了，这两个蛋白就是一定会有这种交联？我这个我不知道，这是 beyond my knowledge 啊。所以就是我就有点担心那个，即使是它再放在一起的那两个蛋白哈，这样。就说这个，因为你就是只要是在一个 tube 里面的，就认为他们这个可能是对的；如果不在一个 tube 里面的，肯定是错的。但我现在就有点担心，这个可能在一个 tube 里面的也不见得是对的。
这话是对的，就这是对，但事实上我们也会普遍认为，比如说 intralink 在大部分情况下都是对的。
对，
这个也是个假设。
不是，大多数情况下都是这大多数情况下是。
对，大多数情况就是说我们。
所以这个的 FDR 的估计是对 FDR 的一个，其实还是个低估。
这话
因为它是
对
一个
的。
FDR， 它可能是有个范围嘛，它可能是对它的那个 lower boundary 进行了一个估计。
这话是对的，就是说，那最好的情况，比如说我 mix 一对儿，然后我就打一个 LCMS， 我看看这一对儿是不是有 interaction， 如果有我才算，如果没有我就不算，也可以这样。
所以这个 FDR 的问题就是个。
对，但是这个基本上是我能想象的比较简单的，当然它不是完美的，因为就像您说的，它不是说放在一块儿就 cross link， 但是像 peptide 其实也是一个道理，你就比如说你放 peptide 去 cross link， 它也不见得会每一对都 cross link。
是用非常非常高浓度。它才能够
才能 cross link，
非常非常高浓度，然后它已经很拥挤了，然后才能够好，我没有别的问题。
请教一个问题，就是说 CMV 的这个 values 啊，它有很多的原来被发现是未知的蛋白，对吧？没有 annotate 出来的。那么这些蛋白在您的这个里边是不是已经考虑进去了？另外，如果说这些蛋白假如说拿掉或者不拿掉，在你形成的你的这个最后的 cross link 以后形成模型当中，就如果说拿掉以后，它会形成好多的 hole， 让你能看得见。这个地方我不知道有没有看过。
这我有好多 data 都没有收啊，就是说有一些 host 的蛋白，就是宿主蛋白，其实如果把它拿掉了之后 ，virus 是不能活的。就是其实 virus 它会用一些宿主蛋白去，比如说我们找到了一个蛋白叫做 PP1 嘛，就是 protein phosphatase 1， 就它会 dephosphorylate viral proteins， 然后把它拿掉之后呢 ，virus 就不太能够 replicate， 因为它会出现一种 hyperphosphorylation stage。所以说，其实很多宿主蛋白，就我们当然只 follow 了一个啊，可能有很多其他的，就是它其实是在病毒里面是有功能的，就拿掉它就是 variant 就不太行了啊。